Let's talk about cells and how things get in and out of them. So we have a cell, and you know that this cell has a membrane. And the membrane is made of phospholipids. So you have phospholipid here, phospholipid here. You have that phospholipid bilayer making up your cell membrane. And this goes all the way around. And occasionally in here you have some proteins thrown in. Well, one of the important functions of the cell membrane is it controls the movement of substances in and out of the cell. So you have your blood, and then you also have your tissue fluid. Tissue fluid is also called interstitial fluid. And substances move back and forth. So let's say, for example, you eat. When you eat food, you get glucose in your blood. That glucose then has to move from your blood into your tissue fluid, and from your tissue fluid into your cell. You breathe, you get oxygen into your blood. The oxygen moves from your blood to your tissue fluid to your cell. Your cells are making carbon dioxide as waste product. That carbon dioxide moves from your cell to your tissue fluid into your blood. So stuff is constantly moving back and forth, and the membrane plays an important role in what gets in and out of the cell. We're first going to look at diffusion as a way for substances to get in and out of the cell. Diffusion is molecules moving on their own. And the simplest way to think of it is just that they don't want to be crowded. So anytime molecules are crowded, they will naturally spread out. And they spread out until they are evenly distributed. And this is called moving with their concentration gradient. So anytime they're going from high concentration, where they're crowded, to low concentration, where there is open space, we say that they are moving with their concentration gradient. So diffusion is molecules moving with their concentration gradient. They simply spread out from where they're crowded to where there's open space. So now let's look at this in the context of a cell. So let's label some things to begin with. Here is your membrane. There's those phospholipids that form the membrane. This area is your tissue fluid or interstitial fluid. So this is the outside of the cell. And the blue area here represents inside the cell. which is your cytosol. The first picture here shows simple diffusion. So we have the red dots represent oxygen, the black ones represent carbon dioxide. You can see here outside the cell there's several oxygen, there's only a few inside. So the oxygen is moving in. The carbon dioxide has its high concentration inside the cell, it has a low concentration outside, so it is moving out. So again, the same rules as diffusion. Molecules are moving on their own from an area of high concentration to an area of low concentration. This is simple diffusion because notice they're just passing between the phospholipids. So this is things such as oxygen and carbon dioxide. They are small molecules and they pass between the phospholipids of the cell membrane. The next one here is another type of diffusion called facilitated diffusion. So it's the same idea. You're moving from high to low. Here's some molecules moving in. Here's some more moving in. They're both moving high to low. The difference is these molecules are either large or they have a charge that repels them from the phospholipids. So they can't go between the phospholipids. So they use a transport protein instead. So these blue things here are transport proteins. You can think of a transport protein as like a door in a cell membrane. And there's two different types of transport proteins used in facilitated diffusion. We can have a channel, which is always open. Or you can have a gate. A gate can open or close. Okay, so let's think for a minute about this idea of moving from high to low until equal. If we have a cell and we have 2 of the molecule in the cell, and out in your tissue fluid you have 6 of that molecule. Well, this area would be the high, inside your cell would be the low. So we're going to move this way, high to low. The next question is how many? If there's 2 inside and 6 outside, how many will move? Well, the answer is 2. If 2 of them move, you now have 2 more inside, so you have 4. 2 left the outside, so you have 4. 4 and 4 are equal. So remember, they move until they are equal to each other. That's how diffusion works. Here's an interesting thing to think about. You exhale. Is there oxygen in the air you exhale? The answer is yes. When you exhale, that air still contains oxygen. So what happens is you have oxygen in the air, you inhale that into your lungs, and it moves by simple diffusion from your lungs into your blood. Well, remember, it's only going to move until it's equal. Once it's equal, whatever's still left in the air comes back out. So the air has a lower concentration of oxygen coming out. It's 21% going in. It's 16% going out. You do absorb oxygen, but certainly not all of it. All right, let's move on to our next one now is osmosis. Osmosis is the diffusion of water. So one of the things to keep in mind of the osmosis is it applies to water only. And what's happening here is water is attracted to a solute. So a solute is a molecule that dissolves in water. So for example, common solutes would be like sugar and salt. Water is attracted to these molecules. It wants to be with them. It wants to go to where they are. In this picture, these black ones, these black dots, are your solute. Out here is your water. So as you can see, there's more solute inside the cell. So the water is moving in there to be with the solute. So osmosis is defined as the diffusion of water to an area of higher solute concentration. This is very important in your body. Your body has osmolarity, and it's very important to maintain the osmolarity of your blood. So let's look at this a little more. You have your cell. There are solutes inside your cell. You have your blood. There are solutes in your blood. They have to be in balance with each other. They have to be the same concentration. So when they are the same concentration, that is a condition called isotonic. Isotonic means that your blood and your cells have the same concentration of solutes. This means that there's no net movement of water. The water that's in your cells stays in your cells. The water that's in your blood stays in your blood. This is what you want. Let's look at another scenario. You have your cell, it has solutes, and it has water. You have your blood. Your blood has solutes and water. Let's say, though, that you put extra water in your blood. So some examples, real-life examples of what has happened here with putting extra water in the blood. There was a radio station had a contest. Remember when those Wii game systems came out and they were the new exciting thing? There was a radio station that had a contest to give away a Wii. And the name of the contest was Hold Your Wii for a Wii. So the point of the contest was you had to drink a bunch of water and then not go to the bathroom. And whoever could do this for the longest, keep drinking water and not go to the bathroom, would win. Well, the lady who won never got a chance to enjoy her Wii because she was dead a few hours after the contest ended. Another thing that happens, babies have actually died from this. Babies under 6 months old should not have water. They should only have breast milk or formula. And there have been cases of parents run out of formula, so they just give the baby plain water, and then the baby dies. You have to be careful not to get too much extra water in your blood. And one of the things that's going on now is you see people say like, oh, you should drink 8 glasses of water a day. And I even see people walking around with these big jugs that they fill up in the morning, and it's their goal to drink all that water by the end of the day. That's not such a great idea. You don't want to give yourself too much water. So what happens when you have too much water in your blood? Your blood becomes hypotonic. So too much water makes blood hypotonic. Okay. Hypo means less or below. So this means then that the blood has a lower solute concentration than your cell, because all that extra water dilutes the solutes that are in your blood. This means now there's more solutes in your cell than there are in your blood. And remember, water goes to wherever there is more solute. So if you now have more solute in your cell than you do in your blood, that water goes into your cell. So you will now have extra water entering your cell. Well, this is really bad for cells to get too much water in them. Your cells will swell and burst. And this is called lysis when this happens, that your cells will swell and burst because they have too much water. And like I said, it can cause seizures and it can cause death if your cells are swelling and bursting from too much water. We have the other end of the spectrum. You have your cell, it has its solutes, it has its water. You have your blood with your solutes and your water. The opposite of getting too much water is having too much solutes. If you are ever lost at sea, you're on a ship and it wrecks and you're floating around in a life raft out in the ocean, hopefully you know that you can't drink the ocean water. If you're lucky enough to have ever been on a beach vacation and you've been out swimming in the ocean and a big wave comes up and crashes on your face and you get a mouthful of ocean water, you know that it is disgustingly salty. There's one thing to like salt, like putting some salt on your food to make it taste good, but the ocean has so much salt, it's just disgusting and gross. It is very, very salty. If you were to drink ocean water, you would have so much salt in your blood that it would make your blood hypertonic. Hyper means more. So your blood would have more solutes than your cell. So remember, water goes to the solutes. So if there's more solutes in your blood, water will leave your cell and go to your blood. So this will cause your cells to lose water. And your cells will actually shrivel up, kind of like a deflated balloon. And when they do this, it's called crenation. And this also can kill you. You can't live if your cells are all dried out and shriveled up. So that's why if you're ever lost at sea, don't drink the ocean water. Drinking ocean water, put too much salt in your blood, make it hypertonic, your cells will crenate and you'll die. One more thing to think about with this. is IVs and contact solution. Next time, if you're in the hospital getting an IV, or if you wear contacts, or if not, just look at this the next time you're in the aisle at the store with the contact solution. Both of these are saline. Saline means salt water. Now, they're not nearly as salty as the ocean. They're just a little bit salty. And the reason that these things are a little bit salty is so that they are isotonic to your cells. When you get an IV, you want that IV fluid to be isotonic to your cells. That way, your cells don't shrivel up because they're losing water if it's too salty, or they don't swell and burst because you're getting too much water if it were plain water. The same with putting fluid in your eyes. Contact solution has direct contact with the cells of your eyes, and you want that to be isotonic so that you don't damage the cells of your eyes. All right, so that is osmosis, and osmosis is very important in the body. Let's move on now to active transport. Active transport is the opposite of diffusion. So remember how in diffusion molecules move on their own from high to low until they're evenly spread out? We're going to do all of that the opposite. First of all, you're going to force the molecules to move. And you're going to do this with what's called a protein pump. So the pink thing in the picture here is a protein pump. And it's going to use ATP. You have to use energy. So here, this yellow circle is your ATP. And you're going to make them be more crowded. So instead of moving from high to low, you're going to move low to high, so that you make the molecules more crowded. Opposite of letting them spread out and be even. So in this picture, look at the blue ones. These blue dots are potassium ions, which potassium is K+. Which way do you think they're moving? Notice out here is the low, in here is the high. The potassium is moving this direction. It's moving in. You also have some sodium ions here. The pink ones are sodium. The sodium ones are moving out. They're moving from low to high. So this is a sodium-potassium pump. Your cells have sodium-potassium pumps. And these are found in most of the cells in your body. They are especially important in neurons. These pumps are continually moving. They push sodium out of your cell and potassium into your cell. So in general, most of the sodium in your body is outside your cells. It's in your blood and your interstitial fluid. And most of the potassium in your body is inside your cells. Now notice I said most, not all. You do keep a little bit of sodium in your cells. You do keep a little potassium in your blood. But these pumps do a pretty good job of keeping most of the sodium out and most of the potassium in. And our final method of transport here is vesicular transport. This is when you have your substance in a vesicle. So you've already learned about the parts of the cell. You know about the Golgi apparatus. So let's say here is the Golgi. You have also your cell membrane. And let's put a vesicle in here. All three of these things, the Golgi, the vesicle, and the membrane, are all made of phospholipid. So they're made as the same thing of each other. So they can just pinch off and fuse together. So vesicular transport goes two directions. You have in and you have out. Let's look at exocytosis. So exo, think of exit. If you're doing exocytosis, you're going to pinch off a piece of the Golgi. And this piece of the Golgi that you pinched off forms the vesicle. And the substance that you're trying to move, so let's put a little substance in here, is inside the vesicle. It then moves to the membrane, and because the vesicle is made of phospholipid and the membrane is made of phospholipid, they simply fuse together. And now your substance is outside the cell. We can also do endocytosis. So think of enter. This is where you pinch off a piece of the membrane. To make your vesicle. With your substance inside. Okay, so let's look at this. Here we have a cell, and here is our substance. Where the substance is, you start to get a dent in the cell there. And that dent grows, so you keep pushing that dent in. And then it pinches off. If you look at this picture going this direction, we have endocytosis. If you look at this picture going this way, if you start, here's your number 1, you have exocytosis. An example of how the body uses this, remember you've already learned about the macrophages that are those white blood cells that eat bacteria? Okay, here's your macrophage. It's this whole big cell. These kind of orange things here, these are the bacteria. Okay, see how the bacteria are in a vesicle now? So right here, you're doing endocytosis. So the bacteria are being taken into a vesicle. You're pinching off a piece of the membrane. These things, remember you learned about lysosomes already. They contain enzyme. They come along to that vesicle with the bacteria, they put the enzyme in there, and they digest the bacteria. So here you can see there's your digested bacteria. Then here, there's that vesicle fusing with the membrane. You're doing exocytosis. And you'd get rid of that digested bacteria. So this is how macrophages are protecting you, and they're killing infection. Sometimes if you have a cut and that cut's infected, you can tell that your macrophages are there working because all this massive amounts of digested bacteria coming out is what you see as pus in the cut. All right, so there's the ways that you get things in and out of your cells. You do membrane transport.